Continuing on from the previous lecture, which was about hemodynamics, we now want to take a look at biophysics of fluid flow in plants, specifically vascular plants. So because we're still on the topic of fluid flow, it kind of makes sense that we should do a recap of fluid flow first, because a lot of the concepts that we've learned in the previous lecture will still apply for this lecture. The next thing we want to go through is just a definition of what a vascular plant is. Basically, there's vascular and non-vascular. It would be good to know the difference between the two of them. And then basically go through how nutrients and water is transported and distributed throughout the entire plant, whether it's from the soil to the leaves or from the leaves throughout the plant's body. So there are two specific types of fluid flow in plants that we do have to learn about that has a strong physical basis, and that's xylem transport and phloem transport. After that, we'll go through a summary as usual. So for the recap, I want to first go through some equations for fluid dynamics, basically all of the important ones that you should bring with you from the previous lecture. Then go through blood composition and circulation. We did make some assumptions about the shapes of blood vessels and everything, so we do want to take a look at that, as well as some important phenomenon that we didn't consider for our modeling. So in terms of equations for fluid dynamics, these are the main ones that you should remember from the previous lecture. First of all, we know that fluid flow, any type of fluid flow, can be laminar, which is smooth, or turbulent, which is basically rough or chaotic. The velocity profile, which was that parabolic velocity profile, it represents the speed of your fluid in a pipe, depending on how far it is away from the center of the axis. So if you want to figure out the speed of fluid flow in a tube, you can use that equation. So based on the r squared minus x squared, the maximum fluid flow would be at the center of your velocity profile, where x is equal to 0. Next, we took a look at Poisson's formula, and that's used to calculate the volume flow rate through tube, as well as resistivity. So we were able to calculate delta V based on that equation and also compare it to an electrical analogy, and we're able to derive an equation for the resistivity of your fluid flow. So knowing that there is some sort of volume flow rate is important. You can just plug and chug using that equation. And of course, knowing that because there is some sort of flow, we do have to consider some other force that can prevent smooth flow from happening, which is R for resistivity. If we assume a continuous flow for an incompressible fluid, the other thing we can do is also apply something called the continuity equation. So basically, this is when you have one tube or vessel flowing into another tube or vessel, or you can have multiple tubes or vessels flowing into a certain number of tubes. And then you can just use N1A1V1 is equal to N2A2V2 and be able to calculate the outstanding parameter that's missing. The last two equations were ones that are more specific. So number one, there's Bernoulli's equation, which we use for one of the examples. It helps us analyze flow energetics. So in terms of this, you have your P, which is your pressure. Then you have some sort of potential energy coming from your flow. That's the PGY, or rho GY for density. You also have some sort of kinetic energy for the speed of the flow, which comes from the 1/2 rho v squared. So just be able to apply that equation whenever required if you're given some sort of kinetic or potential energy that comes from fluid flow. And finally, we had Reynolds number. So it's very different than aerodynamics. It's not over 1,000, but anything over 1,700 means that your fluid flow is turbulent. So RE is equal to 2 rho divided by pi r times viscosity multiplied by the change in volume. So the next thing we talked about in the previous lecture was blood composition and circulation. Of course, this is just a brief overview. Number one, your blood is composed of cells and plasma. and the viscosity of your blood is not going to come from just your plasma. It comes from the red blood cells or the erythrocytes that are inside your blood. So the more blood cells you have, the more friction internally that your blood vessels will have, causing a slower flow rate, or basically a higher viscosity. So the more blood cells you have, the thicker your blood is, the slower it's going to flow. Next, we did talk about the circulatory system, and it is important for you to know where the blood is going if we give you a starting point. So the circulatory system, there's two of them, the lymphs, one for the lymph and one for blood. So we focused on blood, which is made up of blood vessels. And there are two types of blood vessels we focused on. It's arteries and veins. Specifically, arteries are in red and they move away from the heart, and veins return blood to the heart. The main difference is that arteries and veins can carry oxygenated or deoxygenated blood. So taking a look at the diagram here, you have, let's see, laser pointer. So first, I'm going to start with the lungs. We're going to be talking about air exchange in the lungs in the next topic, but you have capillaries in your lungs that are going to absorb oxygen and from your alveoli. So when your blood absorbs all of those oxygen, then it's going to become oxygenated. That oxygenated blood goes towards the heart, towards the left atrium, and then through the left ventricle, which pumps out the blood through your aorta. So that oxygenated blood then gets pumped throughout your entire body, and it branches out into arterioles and eventually capillaries. And those capillaries are going to attach to organs and different tissues in your body to provide oxygen to your system. Once your system uses up that oxygen or takes it away from the capillaries, then your blood is no longer oxygenated, it becomes deoxygenated. So after it's used it up, we change to blue veins, which returns the deoxygenated blood to your right atrium and then the right ventricle. The right ventricle will then pump out the blood towards the lungs again so that the deoxygenated blood can get reoxygenated. So even though we were able to model blood vessels based on those equations that we had at the beginning, so we could use the continuity equation, assume that all of our blood vessels are perfect cylinders with a specific diameter. There are different phenomena in blood vessels that we have to be aware of. So these would be two exceptional cases that I'd want you to remember. First, there's the PHYS-Lindquist effect. That basically states that if you have a blood vessel over here on the left and a smaller blood vessel, potentially a capillary, here on the right, if you have blood cells, red blood cells that are floating inside a vessel, the larger your vessel, the higher the chance that the blood cells can bump into each other and cause increased friction. But the smaller and smaller your vessels get, the more lined up they will become, and that means that they're going to be able to flow a little bit more smoothly because there's less interaction in between multiple red blood cells. So that's that effect. The other effect is called plasma skimming, and that really has to do with the velocity profile. So looking at the diagram below, we have a thicker blood vessel over here with a bunch of red blood cells. Your red blood cells are going to flow more quickly in the center where there's maximum velocity. So there's going to be a higher density of red blood cells in the center and a lower density of red blood cells at the edges of your blood vessel. This means that if your blood vessel does branch out into smaller vessels, then it's going to skim a smaller amount of red blood cells because the majority of your red blood cells are going to be in the center, and you're only left with the low density blood cells at the edges. So that would describe plasma skimming. So now using all of the knowledge that we have from our fluid dynamics chapter, we want to take that knowledge and apply it to transport in plants, the other type of living system that is also quite popular. And we want to be able to talk about vascular plants. So first, going to go through what vascular versus non-vascular plants are, and then we're going to take a look at what types of vascular tissues there are in those types of plants. So there are two main types of plants. There are those that are going to grow close to or on water, and there's going to be those that grow away from damp areas. So basically land plants or flowering plants. Land plants require what's called lignified or vascular tissues. And that's because they need to be able to have a way to absorb water from the ground around them because they're not close to water. So we call any type of plant that requires this a vascular plant. Of course, there's exceptions, but the majority of vascular plants are land plants. Now, non-vascular plants don't need vascular tissues as much because they're found in damp and moist areas. So that means that water is readily available to them. They're not in need of being able to have mechanisms to absorb water since they're always around it. So I have a picture here of what's called an angiosperm or a flowering plant. It's basically a perennial that you can plant in a garden or in your backyard. And the question is this a vascular or a non-vascular plant? So if you see a flower like this and you see soil, most likely it's going to be a vascular plant, and that would be a good assumption to make. So because it's a vascular plant, that means that it needs the proper anatomy in order to take in and process water. Let's see what an example of a non-vascular plant could be algae. So it's always near water, and so therefore its anatomy is going to be slightly different. So from this, as a biophysicist, it makes sense for us to study one type of plant in more detail, and that would be vascular plants, because that means they take in fluid. There's going to be fluid flow, and that's what the rest of this lecture is going to be dedicated to. But before we can actually go through the mechanisms of fluid flow, we should understand the anatomy of these vascular plants first, because they're not going to be the same as the cardiovascular system of humans. Okay, so the first step is to understand what a vascular tissue even means. So humans have the cardiovascular system, and these plants have vascular tissue. The term vascular basically means something that consists of a channel or a tube. In humans, the channels carry blood, and in plants, the channels carry water and nutrients. So in plants, there are two types of vascular tissue. One is called xylem, and the other is called phloem. These two tissues are really closely tied to one another, and pretty much located side by side. And when they're side by side, they get grouped together and are called a vascular bundle. So the job of this vascular bundle is to transport fluid and nutrients internally throughout the plant. So then one good question is, what do these vascular bundles even look like? And I think one of the best examples is a celery stalk. So if you've had celery before, which I'm pretty sure all of you had, cut it in half, and you'll be able to see these little dots in your celery. And each of these little dots or tubes, those are called vascular bundles. Within each vascular bundle, there's xylem and there's phloem. So yeah, you can actually do a fun experiment where you show water transport by adding some food coloring to water and then sticking your celery stalk in it, and it should transport some of the water up and basically make all of those channels colored in whichever color you prefer. Now, the difference between xylem and phloem, the primary function of xylem is to transport water and nutrients up in one direction, while phloem allows for two-way flow over here, two-way flow of water and nutrients. That's because xylem's main function is to deliver water to the plant, whereas phloem's main function is to transport nutrients around the plant. So basically, you have leaves that produce photosynthesis products, and your phloem is going to be responsible for taking those products through a water transport system and transferring it up and down the plant's stalk or roots to different areas of the plant. So those are the two main differences between xylem and phloem, and we're going to go into a bit more detail about each. So what makes up the xylem? Since these are essentially vessels, it makes sense that they're long. There are water conducting portions of the xylem made up of what's called tracheids and vessel elements. And there are also supporting cells such as your parenchyma and fibers. There are differences between a tracheid and a vessel element, and it's important to know the difference between the two. So if we know that xylem is responsible for transporting water up the plant in a one-way flow, there are two ways that the flow can happen. They can happen stacked on top of each other so that you have a really long tube of xylem cells just stacked one on top of each other. If you're looking at a continuous vertical tube with different sections stacked on top of each other, that is what's called a vessel element. So they have what's called perforated pores over here, or a perforation plate between each vessel element, so it allows for vertical flow. So that basically looks like a bunch of cylinders stacked on top of each other vertically. Now, tracheids have pits that allow for lateral flow. So they're not really cylindrical. They're more like oval shapes with really sharp ends. So they're tapered at one end and they taper on the other. And each one kind of fits in next to each other. So I'll draw a little thing for a tracheid. Kind of looks like this. And then you'll have another tracheid kind of attached on the side, and then there's going to be these pits on each side that allow for water transport to move laterally. So those are called tracheids, and between vessel elements and tracheids, it allows for the movement of water up and around the plant. Oh, so back to the slide. If we know what the structure of tracheids and vessel elements are like, we could probably make an assumption for which one we'd prefer to model. Tracheids aren't as easy to model, but vessel elements are cylindrical. And so these would be your, let me put that in blue. So the vessel elements would be more like this, with little perforations for each of these perforation plates. So just looking at these two, it makes sense that from a biophysical perspective, we might want to use vessel elements as our model for fluid flow because it's a cylindrical tube. Now, looking at phloem, which is the other type of vascular tissue, it consists of something called a conducting cell or sieve elements. It also has parenchyma cells, which can be specialized or unspecialized. So you don't have to know all the details about this, but you do need to know that phloem consists of conducting cells, such as companion cells, and they're responsible for transporting sugars and nutrients throughout the plant. So the key thing with phloem is that the conducting cells are living, and they have semi-permeable membranes that are made of living cells. So these are going to be connected to what's called companion cells, which connect the phloem to sources such as your leaves and sinks such as your roots. So now we can move on to understanding what types of transports are possible in plants for nutrient distribution. We already know the structure, so it's time that we understand how those nutrients are carried throughout the plant. And then we can try to model them using the equations that we already know from our hemodynamics chapter. So in this diagram, what we can see is that there are water and nutrients. Let's just get the laser pointer back. We have water and nutrients that are represented in green and blue, and they are usually drawn up from the soil by the roots and transported throughout the plant through the xylem. So keep in mind, Xylem is the water conducting channel in plants that allows for the transport of water upwards. So because it can only go up, the only thing that's at the lowest part of your xylem is your roots. And so the roots are going to absorb nutrients and water from the soil and bring it up to the plant. So these xylem channels can go as high as 200 feet above the ground, which is pretty cool. The other thing is phloem. So organic compounds like sucrose that are produced by photosynthesis in your leaves, these are called the sources. They're going to be distributed through the companion cell into the phloem, and through the phloem, you have transport of those nutrients and sugar elements to go to different sinks. For example, your root cells are going to require nutrients to function properly. So you're going to get sucrose from photosynthesis in your leaf, which is a source. And then the phloem transport is going to bring that sucrose down to your root cells so that they can carry out their functions and have enough energy to do so. So in terms of methods of nutrient distribution, number one, there's transpiration. That's the main process of water moving within plant tissues. So water is usually transpired from the plant to the atmosphere through leaves, specifically through the stomata. This is what causes something called a transpiration pull, basically surface tension. So if you're losing water up at the top of the plant with the leaves, that means you have a lower concentration of water at the top. It causes or it creates surface tension, and then more water is going to be pulled up from the soil. So that's called negative pressure, which we'll go through in a bit. Next, you have absorption. Absorption occurs when your root cells are going to passively absorb water in the absence of transpiration, and this is done through osmosis to create root pressure. The third and final type of distribution or water transport is called conduction, and that basically goes through your phloem and your xylem channels. So it's the transport of sugars throughout your plant's phloem and xylem. And one thing to remember is that conduction occurs always from a source to a sink. So keep in mind that a source and a sink may not be always higher than the other. Sugars are usually produced in the leaves, and those leaves are going to be your sources from photosynthesis. And then through photosynthesis, those sugars can be transported to growing roots, which are called sinks, and they use it to grow and to do their function. Now, if we take a look at the minerals, the source of your minerals and your water is going to be from the roots. So then the roots and the soil is your source. And then your sink is going to be the leaves and the shoots of your plant to allow for cell growth and division. So just keep in mind, depending on whether you're looking at water, nutrients, or sugars, your source and your sink is going to be different. So if we ask you a question on a multiple choice portion of a test, and we state that we're looking at the transport of nutrients that are already in existence in the ground, you should be able to name the source and the sink correctly. Okay, so we have the equations from our hemodynamics chapter, and we know what the structure of xylem and phloem are like. So now it's time to actually look at fluid flow in plants and see if there's a way to model them using the equations that we know. So first of all, we want to ask which equations can we use to describe the flow of water in xylem transport. And so the flow of water, because it's moving water, we call it hydrodynamics. And that is exactly what xylem transport is about. We already know that we can model the vessel elements of xylem as cylindrical tubes. So the majority of equations that we've already established for blood vessels and fluid flow can apply as well. So first, we make an assumption. We can consider xylem transport to be laminar flow of viscous fluid in a pipe. So Poiseuille's law is going to be in effect here, where we have the exact same equation as before. we can also calculate the maximum speed of flow based on the velocity profile. So that's when x is equal to 0. Now with fluid flow, we can also look at how much resistance there is to the flow itself. So looking at resistivity, that's defined as the resistance per unit length. We get the exact same equation as we did before that was presented in the recap. So we can do a sample calculation based on a typical xylem channel of diameter 100 micrometers and a viscosity of water of 10 to the negative 3 Pascal seconds. So R is going to be equal to 8 times the viscosity times L divided by pi R to the 4. And that once you do all the calculations, you'll be able to get 10 to the 5 Pascal seconds per meters cubed. After that, we can compare that to exact results from real life. So if you compare that calculation with some of the statistics, you can actually see that this model agrees well with theory. So some other plants, not so much. For example, in oak trees and in herbs and shrubs, you end up with a much higher resistivity in real life versus what you calculate on paper. So the reason for that would be primarily the sheer size of and the number of branches or fluffiness of those types of trees and plants. So now that we've gone through the velocity of fluid flow and also the resistivity of fluid flow, one thing we've never touched on before, especially with hemodynamics, is a vertical pressure gradient, especially with transpiration. We don't have blood that evaporates from our cardiovascular system, but plants have water that evaporate from their leaves. So we have to take that into effect. So a pressure gradient is defined as the pressure difference per unit length of xylem channels. It's required to maintain xylem flow, and it depends upon the resistivity of a specific plant. So different plants are going to have different resistivities. So if we assume vines, which are very small and easy to model, we can calculate a pressure difference. So it's delta P divided by the length of your xylem channel. You can use PHYS equation, be able to plug in some values using the same assumptions that we did before, and you get 160 pascals per centimeter. So what this tells us, especially because we just showed that vines are a good model and correlate well between real life and theory, we can assume that a hydrodynamic pressure gradient of 150 to 200 pascals per centimeter is necessary to allow for your water to go up in your plants. Anything else might not work. So that pressure gradient is what's going to act against gravity to pull your water up and provide flow velocity. Now, your pressure gradient, where does it come from? So that's a really good question. It can come from two different places. You can have positive pressure and you can have negative pressure. One of them is going to contribute a little bit more than the other. So positive pressure basically means that you have a contribution from your roots, and it's going to help with hydrostatics. So primarily with positive pressure, you're looking at osmosis, whereas with negative pressure, which is the main contribution of your pressure gradient, you have a contribution from your leaves through transpiration of water. So water gets evaporated or transpired through your leaves, and then that makes the concentration of water much lower at the top or at your leaf, and it's going to cause more water to flow upwards and produce surface tension. So that is the main driving force of hydrodynamics in plants. If you ever get a question about which of the following is most responsible for the pressure gradient that allows for hydrodynamics in plants, then you would say it's negative pressure caused by the transpiration of water through leaves. So we're just going to take a look at the differences between positive pressure and negative pressure now. With positive pressure, another term for that is called root pressure. So what's happening here is that your roots themselves are going to absorb a lot of nutrients from the soil, and that means there's going to be a high concentration of solute. And the water potential of your root cells is going to be more negative than the soil, so that your solutes are going to want to move into your root. So that causes something called a positive pressure that forces the sap up the xylem and towards the leaves. The main driving force for this is osmosis. It's the spontaneous net movement of water through a semi-permeable membrane. So it's permeable to solvent, but not solute. Now, the movement is always going to be from a region of low solute concentration to a region of high solute concentration, so that makes sense. Now, if we take a look at negative pressure, which is the most important part that we would want you to know, negative pressure is also called transpiration pull. So in order to understand what transpiration pull is, we have to understand what a capillary force is. So a capillary force is when you have a very, very small tube, and then when you have water inside, it's going to form there's going to be surface tension, of course, which allows water to climb up the sides of your capillary tube. And then that U-shaped curve is called the meniscus. So the surface tension is something that's going to allow water to climb a small diameter xylem tube, which is what most plants experience, because their xylem tubes are very, very small. So what can we say about negative pressure? We can say that xylem, water, and sap flow is caused by the evaporation of water from your leaf cells or your mesophyll cells to the atmosphere. What's happening is that as the water escapes from the leaf, that means you're creating a lower water concentration in your leaf's cell walls. And because of that, there's going to be a rush of water to try and fill in that gap. So before that happens, there's tension in the entire volume of water in the xylem, and then the column is going to draw up more water from the roots so that it can balance things out. So using this knowledge, we can apply that to phloem transport as well, but just keep in mind for phloem transport, your water can go up and down. For phloem transport, it mainly is going to be the transport of sugars from your sources, which are leaves, from photosynthesis, to sinks or roots. The food in your phloem sap, and the word sap just means the water inside a plant that has some sort of nutrient inside it. So the food in your phloem sap can be transported in any direction. So that's absolutely crucial to understand, but it can only be transported as long as there is a source of sugar and a sink for the sugar to be transported to. So if you don't have a sink and a source, then there is no phloem transport. So it does flow at constant speed, and it's primarily run by osmotic pressure gradients versus transpiration. So it moves approximately 1 meter per hour, which is very slow. Based on the osmotic pressure gradient, you should understand how it's created. So number one, your source cells or leaves are going to have a lot of sucrose concentration because of photosynthesis. It's going to develop and create sugars. Water is going to enter your source cells or your leaves through osmosis because of your higher solute concentration. This causes a lot more pressure, and so then your sap is going to flow out because of that extra pressure. What's going to happen is a lower osmotic pressure will be created in the sink or the root because of sucrose withdrawal. Because what's happening is in the roots where the sucrose goes to, it's going to absorb the nutrients, it's going to use them, and so the solute level goes down. So that means it's going to want more solute. So this difference in pressure between the root and the leaves allows you to move fluids with a constant speed from your source to your sink, whether it's up or down. So what about phloem transport? Is there a way to model this? We have to present something called Fick's law. So what we can say is that in phloem transport, the amount of substance or solute that's transported per unit of time is going to be proportional to the tube's area, as well as the concentration gradient along the tube. So the best way of doing this is to use a molecular diffusion model, and one of the most famous models is Fick's law. So Fick's law has everything to do with flux, and flux is basically a rate of flow of something. So the flux, or the rate of flow, is going to go from regions of high concentration to regions of low concentration, just as usual, and is going to be with a magnitude proportional to the concentration gradient. So the greater your concentration gradient, the faster that flow is going to be. So we come up with a few terms. We have D being the diffusion coefficient. We have delta M being the amount of substance being moved by diffusion at any specific time. So if I want to figure out flux, we usually represent this as J. J is going to be equal to delta M over T, the amount of substance moved, or the flow per unit of time. But then we can also represent this by D, diffusion coefficient, times A, which is the area, times the concentration gradient. So that's an equation that we can use if required. Now we have one practice problem for this chapter. We know that because of the forces of surface tension alone, water rises in a capillary. So on the right, we have a picture of a tube. We can assume that it's a xylem tube with a radius of 100 micrometers. So R is equal to 100 micrometers. The height is equal to something that you'll have to solve. So under surface tension, we want to figure out how high the water will rise, try and figure out which equations to use for this, and then we'll take up the solutions in the live lecture. So what should you focus on for this chapter? First of all, you should understand what vascular plants are, as well as their composition, so specifically xylem and phloem. You should know the different mechanisms of water transport in plants, so the difference between xylem and phloem channels, and how they move water up and down, where the pressure is, whether it's positive or negative pressure. And of course, when we're talking about fluid flow in plants, do know about surface tension and capillary forces, just a general overview, as well as Fick's law. We will be talking about surface tension a lot more in the next unit.